I am Erin Williams. I am the MiND program manager. MiND stands for Michigan Neurodegenerative Diseases program. And today I'm really excited to share with you all how types of biological samples can help us understand Parkinson's disease better. And Ted really set up my whole talk very nicely. So we might be able to get through this a little bit quicker now that you have such a great overview from Ted. But the goal of the MiND program is to catalyze neurodegeneration research here at VAI to move the needle toward a better understanding of the genetic and epigenetic underpinnings of neurodegeneration. In other words, that means we are trying to enable and promote research here at the Institute so that we can understand what contributes to Parkinson's disease. And to do that, we have created a few different initiatives that I'm going to go through today. And we're going to talk mainly about how we can integrate biological samples from patient donors to understand the disease mechanisms, very much like what Ted is doing. And so we have created three main initiatives. We have a neurogenomes initiative where we're taking blood samples from maybe some of you and creating a biorepository. Biorepository, also called a biobank, is simply a way that we take and store any sort of biological specimen in an organized fashion, and we store it for a long time for uses immediate or in the future. And I'll talk a little bit about what we're using those samples for right now. We also created a brain bank. So instead of blood, we're collecting human brains. Human brain tissue is really important for researchers because that's where we can see what is actually happening in the disease. It's really important for researchers to be able to get their hands on, so to speak, that tissue. And we've also developed an induced pluripotent stem cell and screening platform. And so Ted set up very nicely iPSCs. And we're basically hoping to integrate samples from both the Neurogenome Initiative and the Brain Bank into this screening platform so we can really try to dig up and take apart the disease mechanisms. And then we also have a small internal funding mechanism to help promote science here at the Institute. We have a lot of really smart people that are involved in this program. The program is led by Dr. Darren Moore, and the Neurogenomes Initiative is directed by Jerry Coetzee, the Brain Bank by Mike Henderson, and the iPSC and screening platform by Laurent. And so this is kind of a figure. I'm going to come back here so I can see what I'm looking at. This is a figure of how we envision these biological samples to integrate to help us better understand PD. And so thanks to the selfless donation of donors, both those with disease and healthy controls, we have started to collect different biospecimens from both West Michigan and around the US, including brain and blood, and then a component of blood is plasma. And today I'm going to talk to you about how we're using those samples and how we envision being able to contribute to a better understanding of disease. And so first I'm going to go through our neurogenomes project. So this is where we collect blood and then try to understand the genetic landscape of disease here in West Michigan. And so the Neurogenomes Initiative is really, the heart of it here is in Grand Rapids. We partner with local clinics, Corewell and Trinity, to recruit patients here in the area that have disease. And then what we do with those is we isolate DNA from all of those samples and we send them to a company called Invitae. Now what Invitae does and what they specialize in is full gene sequencing as well as deletion and duplication analysis. So what they're doing is they're taking a panel of 26 genes that have been associated with either Parkinson's disease or Parkinsonism, and they're looking at the entire length of the gene and seeing if there's any known mutations or any variants that aren't necessarily known to cause disease, but are present in one of those 26 genes. And so far, we've collected 263 samples from people with Parkinson's. 244 of those samples have been sent to Invitae. And interestingly, genes here in bold have been identified in our population here in West Michigan to have either a known mutation that's causal for Parkinson's or Parkinsonism, or there's a variant in one of these genes that doesn't necessarily cause disease, but may be associated and could potentially be a new genetic association with Parkinson's disease. So as you probably know, only about 5 to 10% of PD cases are genetic, but we thought it was really important to understand if our population here in West Michigan represents other similar populations, or perhaps maybe there is something different about this population. Maybe we have a founder effect is what it's called, where people from, the Netherlands, we have a large population of Dutch here. If when they moved here, if they brought a disease that causes Parkinson's with them. And if that's the case, this is how we would be able to find it. And so because we have found some interesting mutations or variants, we're able to take those and integrate them into an iPSC and screening platform. So like Ted mentioned, we can take the blood samples, get peripheral blood mononuclear cells from those samples, reprogram them into iPSCs, and then use those iPSCs to differentiate into whatever brain cell we're interested in, and use that to try to understand disease mechanism. So what is going on in these specific patient cells that's not happening in a control cell? And to do that, we have created this screening platform. So this is kind of just an overview of like what we're trying to do, right? So we have brain and blood samples. Maybe eventually we might get some skin fibroblasts. Those are just a little bit easier than blood to differentiate or reprogram into iPSCs, but generally they're kind of the same idea. We can reprogram them into mutation curing neurons, or we can correct them, like Ted was talking about, into healthy neurons, or I guess that's up here. And we can also look at just healthy controls. And we can use this screening platform that I'm going to tell you about to kind of dissect what's happening. And so in order to dissect that disease phenotype, so phenotype is trying to understand why the cells look the way they do, why maybe one cell has enlarged lysosomes and another one doesn't, for instance. And so to do this, we have a really cool setup called a high content imager. And a high content imager is just very fancy, expensive microscope where we can use these cells and look at different proteins, molecules inside the cell in a very high resolution manner and in a quick manner. And so
is that in the Institute?
Yes.
Yes.
It is one floor down. And everything is connected by a robotic arm. And so here is like what the imager looks like. You would never know that that's a microscope, but that is a microscope. We also have an incubator where the cells can live. We have a plate washer, which is helpful. Like if we're treating the cells in any way, we can wash the cells and then put a treatment on and, switch around the liquid. And then this guy here is a liquid handler. And so a liquid handler can take these plates like in a 96 well plate. So you can have 96 different samples at a time. And it can pipette different volumes of liquid into each of those cells at one time. And so this in and of itself is its own robot. And then this robotic arm can move plates that contain cells from any other component of the system. And so why do we care to do this, right? Why do we need an expensive, fancy microscope? Well, I thought I'd give you an idea of what traditional microscopy looks like, okay? So typically when we're looking at cells, whether we've derived them from blood cells from humans, or we've gotten them from a mouse, or they're an immortalized cell line. Typically what you do is you manipulate your cells in whatever way, whether you're adding a drug or you're doing some genetic manipulation. Treat them manually, one well at a time, using a pipette, and it takes hours. Then you put the cells onto a microscope slide, and then you image the cells, or image the slides one at a time, and then you manually analyze all of your data, most of the time by circling your cells in a program with a mouse, literally hundreds and hundreds of times over. Most of my PhD was done doing this, and it took me a long time. One of the big advantages of high content microscopy is that, one, you can do a lot more samples at one time. It can also be it can help with bias, right? Because we're human, we can make error. I can accidentally add a compound to the wrong well and never know it, right? One thing that high content microscopy can help us with is to get rid of that bias. And another thing is that it's just really set up and go. Honestly, we can press a button and leave the room, and then hours later come back and have analyzed data. So that's something that's really, really valuable to our researchers here. And so everybody loves robots, right? So this is the video of our robotic arm taking a plate from, those are called plate hotels, putting it into a plate washer. The plate washer is then going to run a wash. So it's gently washing these cells, washing away the media or the drug or whatever. And then the robot can take the plate and put it in the liquid handler. So this liquid handler is, again, its own robot. So you get to see two robots in one video. So you see there's the robotic arm bringing our plate, and it's going to gently put it in our liquid handler. And then our liquid handler, its own robots, is gonna take pipette tips from here eventually and put in a new liquid into that plate. So imagine this being a drug we're interested in looking at. So you notice it's doing 96 wells at a time. This can go up to 500 or 15 or 1,536 wells at a time. Well, not at a time, but it would do it in like batches, but it would take way less time. So I think that's pretty cool. You don't often get to see the like nitty gritty details of how a lot of these experiments are being conducted. So I thought it would be kind of fun to show you. And these are the types of images you can get. And I think these are gorgeous. I'm of course biased. But we look at all different cell types, all different types of models. So these are like primary neurons. These are fibroblasts. We actually even have some pictures of rodent brains. These are induced pluripotent stem cells. So just gorgeous, gorgeous images that are very detailed that we can learn a lot about disease from. And then quickly, I'm going to go over our brain bank. And so the brain bank is really important for a variety of reasons that I'll go through. But we really wanted to set up this resource here at VAI because having brain tissue to research is critical. And so we've partnered with two organizations, LifeNet Health and Gift of Life Michigan. Both are organ procurement organizations. So if you have the little heart on your license, those are the organizations that coordinate any sort of tissue donation, whether it be for transplantation or, in our case, research only donations. And so we've collected 122 brains so far, 84 of which have a clinical diagnosis listed here. Yeah.
Just a quick question on the process. So if you sign up for donation, there's nothing else that an individual needs to do to ensure a brain would come here, or do you have to take another step to ensure you would get that tissue?
So a couple different ways. So with LifeNet Health, all of the consenting is actually done after the patient already passes away, and their family will be told about our program. And if they, agree, then the brain will be sent here. With Gift of Life Michigan, we have the opportunity for what's called prospective enrollment. So say you have Parkinson's disease, and when you pass away, you want to be able to donate your brain specifically to Van Andel Institute. We now have the mechanism in which to do that. And essentially, the only thing that has to happen is you send an email to an email address that I'm going to show you in a minute. And then after you pass away, your family, next of kin, will still have to agree to that. However, hopefully they'll know what your wishes are and agree to that as well. And then once that happens, all of the coordination is done by Gift of Life and the brain gets here. So brain tissue is extremely important, as I mentioned, to examine disease-causing changes where they happen. There are actually only 2 other brain banks located in the state of Michigan, both in Ann Arbor. And one brain can actually provide hundreds of samples to researchers. And so far, since we started this program in the beginning of 2021, we have already released over 400 samples to researchers, which goes to show you the power of the brains that we already have. In terms of typical brain banks, 122 brains is nothing. Typically, brain banks have between 1,000, 2,000, 3,000. So we're just scratching the surface. But you can see, based on how many samples we've already released, how valuable this resource is for our researchers here at Van Andel. And we've been able to use this tissue in a lot of different ways. So we've heard about different types of proteins that that seem to accumulate in Parkinson's disease. So alpha-synuclein is one of them. Tau is another one someone asked about. One of the labs is trying to look at how those different pathological proteins happen in the same patient. Somewhere around 80%, 80% of all neurodegenerative disease cases, like Parkinson's, Alzheimer's, have pathology that's not directly related to that disease. And then we're also trying to understand specifically how the tau protein affects brain cells in a couple different models. And then we're also studying how alpha-synuclein actually forms those clumps called Lewy bodies. So those are just 3 very general ways that this tissue is being used. But like I said, with 400 samples being released, there are a lot of other questions our researchers are asking, and they're able to do that because of these wonderful selfless donations. So if you happen to be interested in that, if you want to, after you pass away, have your brain come here as a resource, you just email that email and they go from there. It's really that simple. And so with that, I think a couple people are taking pictures of that slide, so I'll leave it up. Helen, will they get any of these slide decks afterwards?
Yes, and also all these sessions are being recorded, so they'll be available on our website, as all of that is available.
Perfect, so if you miss it here. And so with that, I would just like to thank, first and foremost, donors and their families, without whom none of this would be possible. As well as everyone who makes the MiND program run. There's a lot of people involved and I'm grateful to all of them. And if you have any other further questions, my contact information is here. I have cards too if anybody is specifically interested in contacting me. But with that, I'd love to take questions. I don't know how much time we have.
Yeah, no, you're fine. I think question at the back, and thank you very much, Sean.
I just wanted to clarify what you said. So I had asked about tau before. So again, I'm not a scientist, obviously. I'm just a person with Parkinson's. So tau is also a misfolded protein like synuclein.
Exactly. Yep.
And they're coded then by different genes.
Exactly.
And do we know which genes code for tau?
Yes, the gene that codes for tau is called MAPT, MAPT. That gene encodes for tau and helps make the tau protein.
Okay.
And go ahead.
So I had I did PD generation, and I think it was also run through in Vitae.
Okay, yep, that
So
sounds about right. Yeah, that
do
sounds right.
they test for that gene as well?
So because MAPT isn't traditionally thought of as a Parkinson's disease gene, it's more associated with Alzheimer's. I'm not sure if it's on their panel, but let's see.
And while you're looking for that, again, I know it's kind of repetitive, but like, can you have tau as part of your natural aging process, or is tau always toxic?
Yeah, so actually all of these misfolded proteins,
All of them,
can be a part of normal aging. We think that there may be some conformational differences. So conformation means how those 3D structures are formed. And there may actually be different types or different conformations of the misfolded proteins that may be associated with just normal aging versus disease. And there are a lot of researchers that are trying to understand the differences between those, because unfortunately it is part of a normal aging process. So it's really hard, we have to, when we look at a brain, for instance, right, we have to look at the clinical data that's associated with that donor to help inform what neuropathology diagnosis we give that brain, right? Because somebody as a healthy control donor can give us their brain, right? They had no clinical symptoms whatsoever, but they may have low levels of A beta or tau or synuclein. So it is important to complement both the clinical manifestations of disease or not with what we're seeing in the brain.
and you might not be the person to ask this one too, but with the new, alpha synuclein based tests, wouldn't that be a concern then, that you would have that present and then perhaps never get that disease?
So there's a couple caveats with specifically that the seed assay, basically what people are looking at. So basically they're taking blood, or I can't remember what biofluid they use. It might be CSF. CSF. Taking CSF, using a process in the lab to amplify any of the misfolded protein that's in there, and then comparing that to like controls, right? What we think right now is that patients that don't have any disease have low levels, if any. Whereas if you're clearly disease manifesting and you have alpha synuclein misfolding, that's another key thing, then you would be a positive. It's simply a yes or no in that assay. However, if you have one of the rarer genetic forms of Parkinson's disease that doesn't have alpha synuclein misfolding, like PINK1, for instance, that assay isn't going to tell you if you have Parkinson's disease or not. Another problem is whether or not, as a kind of caveat, we're trying to decide as a field, are those rare cases that don't have alpha synuclein pathology, do we still consider those Parkinson's disease, or do we consider them something else? So, Dan.
Your earliest slide, or you said there were 263 samples so far, but that should be 264, because this morning I went to my neurologist and on the way out.
I'm not supposed to know. I'm not supposed to know. Technically, no, but
Well,
I'm not
anyway,
gonna do anything with the data. I can't tell you anything. Sorry.
I got another question.
A Jessie question.
I just wanted to for anyone who's listening that's not from Michigan, there's the braindonorproject.org you can go to, because I'm from Georgia, so anyone who's not in Michigan, if you go to braindonorproject.org, they'll connect you with a brain bank that's close to you. My dad passed away a couple years ago, and we donated his brain to Columbia University, and it was really, really informative. It was provided a lot of closure to the family. He had diffuse Lewy bodies throughout his brain, including in the cortical region, in addition to tau and beta amyloid and some TDP-43 in his amygdala. So he had a lot of different co-pathologies, and that was, helped to provide closure to the family to know his brain pathology. And so I just wanted to say that brain donor is super important, and I encourage everyone to try to register for that.
I will say, just as a caveat for Van Andel specifically, we do have some limitations because we're not associated with a large clinical university hospital. Unfortunately, the results that we get from patient donors are not going to be able to be communicated to the patient families after the patient passes away, unfortunately. There are a variety of reasons for that. However, what we're hoping to do instead is to be able to provide some sort of aggregate data on a website. Like I said, the MiND program has only been in existence for about 3 years. So we're still pretty young, and we're growing and continually changing and pivoting and, to see what is most impactful for patients. So we hope to be able to provide with the community with some sort of aggregate data site where we can say, this paper was just published using 50 brains from our brain bank, and these are the things that we found. So unfortunately, we're not able to specifically give those results back. Like in your case, that is wonderful, and I'm so happy that you were able to have that for you and your family. I'm sure it brings a world of, closure for you. And I wish, unfortunately, it was a different, but as like I said, as we're growing and changing and maturing, it could definitely be something that we consider down the road.
Hopefully fast question. For the blood samples and tissue sample collection, how do you initiate that process? My dad goes to Trinity for his treatment. How do we make sure he gets added into this, or are you still accepting?
Sure, yeah, we're still accepting. Actually, with Trinity, we're just ramping up. We haven't even collected our first patient yet from Trinity. We just started, or we just finalized, or in the process of finalizing a contract with Trinity. And we're working mostly with Dr. Danette Taylor over there, as well as Jessica Hedeman.
That's his position.
Oh, okay, perfect. So essentially, there are some criteria that we have. It's very loose criteria. As long as your father would be in one of those criteria, like he would be in our inclusion criteria, he will literally just be approached at his next appointment. And if he has time to donate that day, he can. Otherwise, he can come back when it's convenient for him. That's the exact same way it works at Corewell. So we're removed from that. Like all of our clinical sites does they do the consenting and screening for us. Since again, we're not associated with a clinical site. But yeah, that's how it works. So yeah.
All right, that's great. Thank you so much, Erin.